First, let me thank the organizers for inviting me and for putting on this nice symposium. I'm very happy to be here. So I'm going to tell you today about an enzyme that we've been studying, which is called O-GlcNAc Transferase. It's a glycosyltransferase, but a few years ago it was discovered to have a second function. And so we've been looking at that second function, and I'm going to talk today about how this enzyme is capable of carrying out two very distinctly different reactions, one of them being glycosylation and the other being a mid-backbone cleavage using the same active site. Now, many of you know me as somebody who mainly focuses on the bacterial cell wall, and actually my interest in OGT grew out of my studies on bacterial cell wall synthesis. And so I'm interested in peptidoglycan, among other things, and that's a polymer of carbohydrates which consists of a chain of disaccharides hooked together by peptide cross bridges. And the glycosidic bond here is a glycosidic bond between a GlcNAc and another sugar, and that's catalyzed by an enzyme called MurG. And we started studying peptidoglycan synthesis a number of years ago, and one of the first things that we did was to crystallize MurG, was to develop methods that allowed us to sort of study it, but then crystallize it. And so I was hoping when we got the crystal structure that it would turn out to illuminate something sort of broader about glycosyltransferases, and it did. It turned out that MurG was a paradigm for a glycosyltransferase superfamily. It was actually the second member of the superfamily to be crystallized. The first one was an outlier in the sense that you couldn't tell from the sequence that it was related to anything else. But with MurG, there's a sequence motif in the protein that allows one to see that it was actually related to a large number of other glycosyltransferases. And so this superfamily, unlike the other major superfamily, does not use metal ions for catalysis. Instead, it binds the nucleotide sugar in it by a different mode, and I'll talk about that a little bit later. And you can see that it consists of two catalytic lobes, and they both have alpha-beta folds. And in fact, this fold is a classic Rossmann fold, which was discovered by Michael Rossmann, and it's characteristic of proteins that bind NAD and other kinds of diphosphate-containing nucleotides. So when we got this structure of MurG, I was interested in the implications of the structure, but I was also interested in seeing if we could come up with strategies to find inhibitors. And so as I began to think about what we wanted to do, I realized that it would be useful to have another glycosyltransferase so that we could test the generality of some of our screens and other things, as well as selectivity of inhibitors. And so I decided if we were going to do that, then we needed to find a eukaryotic glycosyltransferase. And so I set out to identify what I thought was the most important glycosyltransferase in eukaryotes that used UDP-GlcNAc as a donor and appeared to be structurally related to MurG. And so that was how I came across OGT, O-GlcNAc Transferase. And so this is an enzyme which, which was discovered by Gerald Hart at Johns Hopkins in the 1980s. And as you'll see, it's a pretty biologically interesting glycosyltransferase. And so that was appealing to me. And you could tell by looking at the sort of C-terminal part of the protein that it appeared to be related to MurG, and it also uses UDP-GlcNAc as a donor. Now, it has some features that are different from other GTB family members. Notably, it has a long polypeptide right between the two catalytic lobes, and it has a very long N-terminal region, which is made up of tetratricopeptide repeats. So those are 34 amino acid helical hairpins, and they're involved in protein-protein interactions, and this region of OGT plays a pretty important role in how it chooses its substrates. Now, this protein is actually found in all metazoans, and so it seems to be a hallmark of multicellularity. It's essential in mammals and in most other organisms where it's been looked at. It's critical for development, and so those features make it biologically interesting, but it also is kind of odd for a glycosyltransferase. Most glycosyltransferases in eukaryotes carry out, they work in the Golgi ER, and they are part of assembly lines that make very long oligosaccharides that often end up on cell surfaces and are relatively stable. OGT floats around in the cytoplasm and nucleus. It puts on a single sugar, and that sugar at the sites that it puts something on, and then that is taken off in a dynamic fashion by a glycosidase called OGA. And so that dynamic cycling is more reminiscent of something like phosphorylation and dephosphorylation than it is of classic glycosylation. And OGT, in fact, is proposed to play a role in a wide variety of signaling pathways. And what people think is that in part it is acting as a nutrient sensor. And so as glucose levels go up in cells or around cells, the cells take them up, and a portion is stored, some is burned, and then a small amount goes into this pathway, the hexosamine biosynthetic pathway, which culminates in the production of UDP-GlcNAc. And so the more glucose you have coming into the cell, the more flux through this pathway, the higher the levels of O-GlcNAcylation you have on proteins, and that can lead then to a lot of changes in the cell. And so there can be very widespread transcriptional changes if you have prolonged hyperglycemia. Now, OGT affects a lot of different cellular processes, and that's one of the things that makes it challenging to understand. It's believed to modulate some kinase signaling pathways, and it's thought that it can do that by competing with kinases, serine threonine kinases, for the same site, but also by glycosylating different sites so that you can get more than a sort of a binary pattern on the cell, giving more complex responses. It's also important in regulating proteasomal degradation and protein stability. Many transcription factors are glucanacylated, and so it seems to play a really important role in affecting transcription in some way through glycosylating transcription factors. And it's also been found on both on histones, but also as a component of other complexes that modify chromatin. And so it's believed that it is also linked to the epigenetic program of the cell. And it's been linked to various diseases, notably diseases that involve aberrations of glucose metabolism. So prolonged hyperglycemia leads to diabetic complications, and it's thought that some of those complications are mediated, at least in part, through enzymatic O-GlcNAcylation. It's also been, OGT is upregulated, and levels of O-GlcNAcylation are very high in some cancers, notably prostate cancer. And people have believed that there's a link between OGT activity and the cancer cell reprogramming, and so there's an interest in exploring that avenue. Now, my interest in it, as I mentioned, started a long time ago, around 2000, when we had gotten our MurG crystal structure, and so a lot of this was not known, but it was clear that it was going to be an interesting protein, and so we set out to obtain a structure of OGT. And the work I'm going to talk about today, some structural and mechanistic work, was done primarily by three people, by a former postdoc, Xiaoyang Zhang, who is now an assistant professor at Wisconsin, Mike Lazarus, who was a former graduate student who's now starting his independent career at Mount Sinai, and a current graduate student, John Janetsko. And so we set out to obtain an OGT structure, and our first challenge was learning how to overexpress the enzyme, and that took quite a while. And so many years later, in fact, in 2011, we finally published the crystal structure of this protein, and by that point, a lot of time had gone by. And we were lucky that, since other people were also working on it, that it just turned out to be that you had to have exactly the right construct. But by the time we crystallized it, a structure had come out of most of the tetratrichopeptide repeat domain. And so our structure has 4 TPRs. Human OGT has 13. The other structure had 10. And so we can build a complete model with our structure and the TPR domain structure. Now, if you look at the structure, you can see this alpha-beta fold. This is the N-terminal lobe, and then the C-terminal lobe right next to it. And in purple is where the UDP binds. And so in these kinds of proteins, the sugars typically bind in the C-terminal lobe. And this intervening domain that I mentioned as being unusual in OGT packs up against the C-terminal lobe. And it's very high, dense in positive charges, and it's still not clear what its function is. So it's only found in OGT, and there's literally no information about what its biological roles might be. And then the TPRs wind around the top of OGT of the catalytic site, and in part, I think, help regulate access of substrates into the active site. Now, this structure has a peptide bound as well, and it binds across the top of the two catalytic lobes. And if you're looking down from the TPRs into the active site, you can see the UDP in purple with the peptide lying right over it. And one of the things I liked about this peptide is that it has 3 cysteines and a threonine in it. 3 serines and a threonine, but only one of those serines is actually glycosylated, and that's the one that is pointing right into this cavity where the GlcNAc would be if this were a UDP-GlcNAc structure. And so I think that speaks to the relevance of this structure. And when we got this structure, it was pretty apparent that the sugar had to bind first, because you can't access the active site if the peptide is already bound, but also the sugar helps create the binding site for the peptide. And so it had been proposed to have a different mechanism, and so we did some kinetic studies and showed that it actually binds in an obligatory order where UDP-GlcNAc comes first, and then the peptide comes on, and then there's a reaction. And so when we got this structure and we did the kinetics, what we really wanted to get was actually a more complete view of the reaction mechanism, because you can get a lot of information from a structure, but if you don't have the substrates and products bound, then what you learn is somewhat limited. And so we had a structure with UDP, and we also had a structure with UDP and a peptide bound over it, but that's not even on the reaction cycle. And so we wanted to get product and substrate complexes, ternary substrate complexes, and also product complexes, and that can be a challenge, particularly for the substrate complexes, because obviously they react. But Mike Lazarus, who was a very talented structural biologist, set up crystals with the substrates and found that he got beautiful 1.8 angstrom structure of the product complex when he did that. And he couldn't get it if he actually made the glycopeptides separately and set them up. And so you can see the sugar nestled down into the pocket in this. And then to get the substrate complex, we needed to find a way to prevent reaction. And so we made use of an inhibitor that this guy David Vocadlo had reported in the literature, which has a sulfur in place of the ring ether oxygen. And that sulfur is what glycosyltransferases go through a sort of a transition state in which is dissociative, in which you have a lot of positive charge buildup on the anomeric carbon, and so they have oxonium ion-like character, which is stabilized by the ring ether oxygen, and if you have sulfur, it's not very effective at doing that, and so it's resistant to both glycosylation and hydrolysis. And we set up crystals with this substrate and with the wild-type peptide having a hydroxyl here, and they reacted. So we got a glycopeptide in the crystal anyway, and the good part about that was we could compare the sulfur and oxygen analog and see that they were identical, and the bad part was we wanted a substrate complex, and so we took out the hydroxyl of the serine, and we were able to get a substrate complex, and so you can see it very nicely here. And so I'm going to play you a little movie. It's actually not really a movie. We're just toggling back and forth between our substrate complex and our product complex. And I kind of want you to pay attention to what happens as we toggle back and forth. Okay, that was exciting, right? So people put up these beautiful pictures, and we see these two, the starting state and the final state, and almost nothing happens. And actually, I was surprised by how little happens. And so the question you might wonder about is, what did we learn? And so one of the things that we learned, so if you look again at the motion, what happens is the sugar moves quite a bit, and this acetamido group actually rotates so that when the bond breaks, it actually undergoes a rotation. But otherwise, the peptide and the leaving group are in almost exactly the same place. And I have to confess that I sort of had thought, because people talk about the attack of the nucleophile and then the leaving group leaving, that it would be this dynamic process where this thing comes in and it forces the other guy out. But what really happens is that this bond breaks and the sugar moves away from the leaving group. It's called an electrophile migration mechanism, and moves up. So it undergoes a rotation upwards, and it basically bumps into the nucleophile. And so this is sort of the feminist reinterpretation of what really happens in one of these reactions. at least in an enzyme. So that was one of the things we learned. And then to sort of tell you what else we learned, you have to kind of know what people thought glycosyltransferase had to have. And so they're pretty simple enzymes. And so one of the things you have to have is you have to have something to stabilize the leaving group. And that could be a metal ion or it could be something else. And the thought was that you need to have something to deprotonate the incoming nucleophile. And so if you look at the leaving group, you can see that this phosphate oxygen, I've drawn 2 hydrogen bonds, and those are from the amide nitrogens at the very end terminus of this alpha helix. And so they anchor this phosphate oxygen right near the helix, and so there's very precise positioning because of them, and then there's a helix dipole effect. And so that is a sort of the way that Rossmann folded proteins help bind nucleotides without making use of metal ions, and so that's what this enzyme is doing here. And then in addition, there's a lysine which makes a contact to another phosphate oxygen. Now those are present in the substrates as well as the products, and so they get the sort of reaction poised to go. And then when the reaction occurs, this rotation of this acetamido group forms a hydrogen bond also to the leaving group phosphate, and so it participates in the reaction by helping to stabilize this state when this leaves. And in fact, we've looked at acetamido at a variety of different sugars, and you have to have a C2-acetamide or you can't get reaction. You can change the stereochemistry at various positions, but you need this to be present. And so the other thing we kind of wondered about is why doesn't the sugar just react with water? And so we had, because it is poised to react, and in fact there's a water molecule right above the plane of the sugar. And so the thought had been that maybe glycosyltransferases exclude water from the active site, but they don't. And I think a big part of the answer to that question is that there's a close contact between the amide that is part of what would be the reactive serine hydroxyl and this phosphate group on the nucleotide sugar, and that pulls this substrate in so that it's pretty well aligned, so that when this bond breaks and it begins to rotate, it literally does bump right into the nucleophile. And it may be that this also helps activate this pyrophosphate a little bit more. And so the substrate interactions are very important in getting this reaction to go. Now, the other thing we found is that there is no side chain in the active site to serve as a catalytic base. And so we looked all around, and another paper came out where they also looked all around for side chains that could act as catalytic bases of some sort, and couldn't find any. And so in this other paper, they proposed that this phosphate oxygen was helping to deprotonate the nucleophile as it approached. And we had rejected that sort of idea in part because of this hydrogen bond and how important we felt that it was to the reaction. And it would obviously attenuate the basicity of this considerably. But what we noticed was that in the active site, there were these ordered water molecules, and they're always present in all of the complexes that we've looked at. And one of them lies right above the sugar, and then the other lies above that, and then there's an aspartate a little bit above and behind the second sugar. And so from looking at this, we wondered whether or not we had this aspartate, which is pretty far removed from the immediate vicinity of the active site, acting as a remote proton acceptor. And so there's something called a Grothus mechanism where you can get sort of proton hopping along chains of water molecules as a way of shuttling a proton pretty rapidly out of an active site. And so we mutated this to alanine and asparagine. And when you do that, it impairs catalytic activity. And so that would be consistent with this. And we have a crystal structure of both of these mutants, and they're basically identical looking to the wild type. So the impairment in activity isn't due to a structural change. So there's a little bit of, so we were sort of interested in that mechanism, and we're thinking about doing some kinetics somewhat reluctantly, but gearing up to do it. When all of a sudden we learned that OGT had a second mechanism and decided to divert our attention from kind of fundamental enzymology related to the mechanism of how this was getting deprotonated to looking at this second mechanism of OGT. And so in 2011, this Winship, Her, and another group published papers, came out around the same time, in which they reported that this protein HCF1, stands for host cell factor 1, which is a chromatin-associated protein. It's found in a lot of chromatin complexes. It's also found in a lot of transcription complexes, that it needs OGT in order for it to be proteolytically matured. Okay, so HCF1 was identified initially in the context of viral infection, and that's why it's called host cell factor 1. It's like OGT, it's another essential protein. And it's got right in the middle of it, so it's a big protein with over 2,000 amino acids, and then right in the middle there are 6 repeats of about 26 amino acids each, and it has to get cleaved at one or more of those repeats. And so there's a cleavage site, which was reported to be cleavage between glutamate and threonine. And so you can see that there's a CET site here, and it was reported to be cleaved there. And then there's a threonine-rich region in the protein, which was proposed to be how this HCF repeat, as it turns out, binds to OGT. And so what people had found is that in order that HCF1, if you don't get the cleavage event, you have mitotic defects. And so you really have to have this cleavage occurring in order to get all the way through mitosis. So for a number of years, people were looking for what the protease is that is cleaving HCF1. And they couldn't find it until these papers came out proposing that it was possibly OGT. And so the Winship Her's paper went a little bit further than the other paper in suggesting that it might actually be acting as a protease, although he wasn't sure because this protein is also glycosylated. And so there was this notion that maybe what was happening is you were getting glycosylation of HCF1 and there was some kind of an autocatalytic process being caused by that process and by binding to OGT. And so it was all pretty murky, and it was quite a surprise to people who work in the OGT field because nobody had ever reported that OGT cleaved anything before. And so many people didn't believe it. And what we decided to do was to take a segment of the HCF1 that was not glycosylated, if we could find it, and see if it gets cleaved. And so we expressed the 3 of the repeats, and we found that if we mixed it with OGT, basically nothing happened. OGT and UDP, nothing happened. If we put in UDP-GlcNAc, we observed cleavage. And so that was pretty striking, because it, of course, suggested that you actually needed to get a reaction occurring. And we did another experiment where we took the UDP-5S-GlcNAc, which we know binds in the active site just like the normal sugar nucleotide, but does not glycosylate readily. And when you do that, there's no cleavage. And so that then suggested we really needed to have a reaction occurring. And if we did some other controls. And so everything was consistent with cleavages done by OGT. It requires UDP-GlcNAc, but there isn't any GlcNAc in the starting material or the products. And so, since we didn't know what to do, we just did another experiment, in this case a mass spec experiment, and really we were thinking that we would confirm the cleavage sites. But what we found was that if you assumed that cleavage was occurring between glutamate and threonine, none of the cleavage products had the right mass. If you moved the cleavage site by 1 amino acid, we found that one of them, the very N-terminal cleavage fragment, which ends in a C-terminal cysteine, was a perfect match, and all of the others were off by 18. And so 18, that's water, and so that suggested that we were losing water somehow from our second product. And so One of the ways you could lose water is through formation of a pyroglutamate, where you get a condensation between this amide and the side chain. And we tested that hypothesis by making, we first mutated our peptide so that we mutated out the essential glutamate in the cleavage sites in 2 of them, so there was only 1. And then we ran a reaction, got cleavage products, and then treated them with this pyroglutamate aminopeptidase, which specifically removes N-terminal pyroglutamates. And when we did that, we ended up getting the products that corresponded to cleavage right here, so between glutamate and threonine. And so when people initially identified the cleavage site for this protein, the way they did it was by isolating C-terminal fragments of HCF1 in cells and doing Edman degradation. And so, of course, if you have a pyroglutamate at the N-terminus, that's not going to work. And so they were detecting some amount of product that had this modification removed. And so it was kind of I guess an honest mistake. But in any case, this was a clue, because what it told us was that if a pyroglutamate is forming, then we have to have something happening in this reaction where the glutamate side chain is getting activated, because you obviously aren't going to be able to simply condense this onto this carboxylate. So you have to activate that by some mechanism. And so the next thing we did was we decided that we would try to crystallize the HCF1 repeat with OGT. And we used the 5S-GlcNAc substrate, which is stable. And when we did that, and I was quite excited about trying this, because there is no structural information about how OGT recognizes any of its substrates except for the work that we've done on HCF1. And so this was the first structure which showed how peptides can bind to the TPRs. And I think that I'm not going to talk about it, but I think that what we're learning about the recognition of this peptide probably applies to a subset of glycosylation substrates, but there are presumably other binding modes for how OGT can recruit different kinds of substrates, and so we're focused on that. But in any case, when we got this structure, and we did an overlay of the 5S of the different complexes, our glycosylation complex, which is shown in this salmon color, and then this HCF, where we've replaced this glutamate with a glutamine. What we found is that even though the amino acid sequences are really different, these are basically superimposable in this region of the polypeptide chain. And so here is where the C beta of alanine is, and if this were a serine, there'd be a hydroxyl right there, and it basically overlays with the C beta of this glutamine. And so if this was a relevant structure, we predicted that we should be able to change that glutamate, or in this case it's glutamine, but glutamate to a serine, and it would glycosylate. And so this is our cleavage, our 3R with our glutamate, with our 1 repeat that is functional, and it cleaves. If we replace it with serine, it's completely dead. But if we look at glycosylation, the glutamate doesn't glycosylate appreciably at any site, but the serine does. And so by changing one amino acid, we've now changed the nature of the post-translational modification that this protein is catalyzing. So it goes from cleavage to glycosylation with that one change. And so with the similarity between how these substrates were binding and all of the other information led us to a simple hypothesis, which is that we're forming a glutamyl-sugar intermediate in this reaction. And so in order to get cleavage, we first go through this kind of an intermediate. And so it's a simple hypothesis, but of course it's always better if you have evidence, direct evidence, apart from, not just inferences. And so we wanted to see if we could actually detect that intermediate. And then try and figure out what the rest of the reaction was doing. And so to do that, we had been working with these substrates, which were about 100 amino acids long, because you can see cleavage on a gel. But if we wanted to look for intermediates, we planned on using mass spec. And so we had to figure out what the shortest length of this sequence was that would be cleaved for mass spec. And so it had been reported that, the 26-mer, and indeed we found that as well, does not undergo cleavage. And people had only worked with really long substrates, and we'd worked with this long substrate. And so we were a little bit surprised when we decided to embark on a making a series of things that were 5 amino acids longer sequentially, to find that the very first one we made actually got cleaved. And so all you have to do is add a few more residues here, and it's actually a cleavage substrate. So we had this cleavage substrate, and it undergoes cleavage. This is just a chromatogram where we're looking at the extracted ions. And over time, over about 3 hours, it goes to completion. So it's quite slow. It's a little bit faster for the natural system, but not that much faster. And we can talk about, why that might be. But in any event, this is the starting material. And you can see that as you progress, you see these 2 little blips, and those are our products, and they accumulate, and eventually it goes to completion. But we found that if we actually went looking for some glycopeptides, we saw these 2 little tiny blips, okay? And these 2 glycopeptides, if you follow them as a function of time, so they both have exactly the same mass, but one of them accumulates early and then decreases until at the very end you can't see it anymore, and the other one accumulates more slowly and never decreases. And so one of them behaved like an intermediate and the other behaved like a byproduct. And so we speculated that perhaps the byproduct was getting glycosylated on threonine, because that's sitting there in the active site, and if it did that, it would prevent cleavage, and so you would end up with a permanent glycopeptide that didn't progress. And so we wanted to get rid of that. And so, of course, we could make a mutant, and we did order a mutant peptide. But in the meantime, we thought we would go back to trying our D554N mutant. So if you remember, that's the one that doesn't glycosylate. And so you can see here, this is wild type glycosylating on serine, and I already showed you that. The mutant does not, it's impaired for glycosylation. But if you look at cleavage, it's cleaving. In fact, it looks like it might be a little better than wild type. So it makes sense that it perhaps doesn't need to have that function of shuttling a proton out if cleavage is involving, let's say, glycosylation on glutamate, because there isn't a proton. And so that could make sense. But so we then did a single experiment where we looked at a long time point, and we found that it actually goes to completion faster than our wild type enzyme cleaves HCF1, and we aren't building up byproduct. And so that was good, and we were happy about the byproduct. We didn't really expect it to be faster. And so we went and looked at a much shorter time point and found that within 3 minutes we were seeing product. But we had a huge amount of what we had identified as our intermediate glycopeptide. And we also see another intermediate which corresponds to starting material with loss of water. And so if you follow as a function of, if you carry out a series of experiments at different time points, you can see that the glycopeptide forms first and quite rapidly in the mutant, and then it breaks down and you begin to see this other intermediate. So this precedes the second intermediate. And so then the question, of course, is what are these 2 intermediates? And our hypothesis was that the glycopeptide intermediate was glycosylated on glutamate. And so we went up to Bruker because Harvard didn't have anything an instrument that was capable of doing ECD type mass spec experiments in sugars fragment very easily in mass spec. And so we needed to actually see the fragmentation patterns of the glycosylated peptide in order to localize the modification. And so we did that. And in fact, it turns out that it localizes to the glutamate. And so this is glycosylated on the glutamate. And then the second intermediate is an internal pyroglutamate, we think, which forms as a result of attack of this amide bond onto this carbonyl, getting rid of the sugar. And so here's our mechanism. The glutamate comes in and attacks. We form intermediate 1. We then form our internal pyroglutamate, and those hydrolyze. In fact, they're known to spontaneously hydrolyze to give you cleavage products. And so that is how we think this is catalyzing cleavage of HCF1. And so one question is, why would it be that nature would do this, right? And so it's a pretty slow reaction, and so you might go, well, is it physiologically relevant? And as near as anyone can tell, it really is physiologically relevant. 50% of OGT is bound to HCF1 in the cell, and you have to remember that OGT is really not a protease. It happens to be catalyzing this cleavage reaction, but that's not its primary job, and it doesn't have to do it very fast. It has to do it fast enough so that you can get enough cleavage on the time scale of the cell cycle, and it's certainly fast enough for that. So, and so what people have suggested is that maybe this UDP-GlcNAc dependent cleavage mechanism is a way that you can link, sort of tune the cell cycle to nutrient conditions and modulate them a little bit so it can go faster or slower depending on nutrient availability. And that at least is a hypothesis that one can begin, that one can test. And so we'll see. But one of the questions that we have been, we were kind of very interested in this mutant because it actually glycosylates so much faster than the wild type. And so you might go, well, why is it going so much faster? And I think looking at the crystal structure gives us some clues. And so this is the crystal structure of the UDP thiosugar with the peptide bound over it, the HCF peptide. And this is D554. And so in order for this to react here, you have to get it sort of a little bit closer than it is. And in order to do that, pretty much anything that you do with this side chain brings it closer to this aspartate. And so if you have a glutamate and an aspartate, it's pretty unfavorable to bring them close to each other in an enzyme active site. And so I think that is a key part of the reason why it's so much slower in the wild type. And when you alleviate that, it just goes 2 orders of magnitude faster. So I like biology because it's always surprising, and I wouldn't have thought when I started working on OGT that it would end up having this interesting second function. And I kind of wonder how many other cases are there that are like this that might be out there. And so both of these, so we've shown that they both uses the same active site, basically uses a lot of the same machinery. You're getting glycosylation on serine threonine in one case and glycosylation on glutamate in another case. Now for a nucleotide sugar glycosyltransferase, that's unprecedented, although I should point out that there is ribosylation on glutamates and aspartates. And then you get this rearrangement, and we think that both the glycosylation and the rearrangement happen on the enzyme. We don't know about the ultimate cleavage. And one of the things that we're particularly excited about is having this mutant, which allows us to accelerate cleavage but attenuates glycosylation, means that we can begin to now go into cells and start to try and tease apart some of the effects of OGT. And so one of the questions that we're particularly interested in is, why is this protein essential? Now, it has a lot of functions. It's a scaffolding protein, or at least it hasn't been looked at systematically, but there's evidence in the literature that it acts as a scaffold. It's found in a lot of complexes which are relatively long-lived. And so if you can figure out what some of those interactions are, you can begin to change OGT in ways that allow you to disrupt them. And so that you can ask the question, how important is that role in scaffolding? And you can also perhaps begin to tease apart, is it essential because it needs to cleave HCF1? Are there essential substrates that get glycosylated and have to be glycosylated? And so it's a problem where there's a lot of complexity, but it makes it interesting. And so we're pretty serious about trying to figure out how it interacts with substrates, and then also figuring out how to go into cells and do some of the eukaryotic genetics that we need to do so that we can dissect this problem, because anyway, it's fun. And so I want to thank my group. Actually, most of them, there's a subset of them shown here, because not all of them wanted to go curling. I don't know why. Yeah, I had the other picture that was put up was my group at the Secret Santa, and this is my group in like snowy Boston inside. But anyway, because we don't have a really pretty beach, and so I can't put up a picture of them in front of the beach. But The people who actually have contributed a lot to this work are all shown here. Ben Gross actually started the project when I was at Princeton, and then I mentioned these three, and they've been just great. And I want to thank Sunya Trauger, who was here, I believe, as Scripps, and who was invaluable in helping us to do some of the mass spec that I just talked about, and also Bruker, Chris Thompson at Bruker. And she sort of set us up with that collaboration as well. And I want to thank Winship Her at Lausanne for a nice collaboration, and David Vocadlo, who gave us the UDP-5S-GlcNAc, and Peter Slees, with whom I have had various collaborations in crystallography. And in the case of OGT, his assistance was invaluable because there were some hard aspects of this crystal structure. And of course, the NIH for funding this work. And thank you.